Hello, good afternoon. Thank you for coming and for joining us on Zoom for this third talk in the microbiome and health, the effects of the gut microbiome on health. This is the third talk today, so we are in the middle of the course. Today's speaker is Dr. Bikash Sahay. He's a veterinarian by training, but has turned into a basic scientist. He likes to do research. He did his DVM, or the equivalent of veterinary medicine, and his PhD in India. And then he did 6 years of postdoctoral research at Albany Medical School in New York before coming here to UF in 2012 and joining the faculty in the College of Veterinary Medicine. His research has been on the beneficial use of gut microbiota. He has published over 42 papers and book chapters on different aspects of microbiology and immunology. He also holds 8 patents based on his research at the University of Florida. His work has been funded by the NIH and by private institutions. Today, the topic of his talk is harnessing gut bacteria for the development of preventive and therapeutic medicine. Dr. Sahay.
So can you hear me okay, right? Okay. So first of all, I am very happy to be here. It's the first time I'm arriving in this building and this community. And it feels like very different than the rest of the Gainesville, looks like. And today I'm going to talk about gut microbiota and how we have used it for trying to develop a medicine out of a common bacteria. That is normally every day we is working. Okay, every day we eat in yogurt. So let's talk about what it is gut microbiota. We know that total microbiota, when you talk about it, is all the bacteria that lives on us. So normally there's so many bacteria in our body that if you count them by gene, so there would be 100 times more bacterial genome than our own genome. So we are more bacterial than a eukaryote and human living being. And when you look at a different kind of bacteria that lives in different compartment of our body, we divide the microbiota in depending upon where they live or where they are. So like we have gut microbiota, oral microbiota, skin microbiota, urogenital microbiota, and even in the lung, you have some bacteria that helps you keep going. So initially we thought that we are very sterile, other than very few things we always thought of dirty. But actually we are more dirty than we thought before. So when you look at the total bacteria in our body and look into this pie chart, so almost 30% or little more bacteria that is available or present in our body, they are in our gut. And that makes sense because we know that when we eat, we have tons of bacteria in our gut. They digest the food. They produce a lot of metabolites for our body. And that actually very, very important. And I will come to you like in a few minutes how they are beneficial. Oops, I went other way. So when you look at our gut, how they look like. So if you think about our stomach and this is our rectum. So there are different compartments in our intestine. And depending upon which part of the intestine we talk about, the composition of bacteria and the way what they do, they differ. So in the stomach, pH is very, very low. And that's why very less number of bacteria you'll find. Only 10 power 1 to 3, 10 to 100,000, only few bacteria in the stomach because it's very adverse environment. But as it goes down, you will see the number of bacteria increased. So the most number of bacteria they're found in the colon. There are tons of bacteria there. And which kind of bacteria they live? First of all, you will have most bacteria, they are there, they can survive without oxygen. There are a lot of anaerobes will live there. And the major bacteria that we are already listed here, like Lactobacillus, Streptococcus, Staphylo, Enterobacteriaceae. These are bacteria. Enterobacteriaceae is a bacteria that lives in the gut, like Salmonella, E. coli. We have heard of that. Those kind of bacteria. But there are some beneficial bacteria as well. And some bacteria, we also use them as a probiotic, like Bifidobacterium is one of them. Bifidobacterium, Lactobacillus, Bacteroides. These are common bacteria that you will find in the pill, a probiotic pill. And these bacteria do a lot of different things. So what they do is they can compete with the infectious agent. This bacteria will fight for the food and space to survive in the gut, so that infectious agent cannot survive. They breaks down the complex carbohydrate. We heard of like sugar, simple sugar versus complex sugar. So these are the one who is going to break down your complex starch and fibers and use them as a food. And in return, they will provide useful metabolites that help our gut health. And then they also produce a lot of vitamins like vitamin Ks, vitamin Bs. SCFA is short chain fatty acids. Like acetate, lactate, butyrate. These are small molecules. They are just produced after fermentation. And these are the molecules our immune cells needs them for epigenetic regulation, also needs them to calm the inflammation down. So that's why the bacteria plays a very, very essential role. Then also what they do, when you have a bacteria in the gut, right bacteria in the gut, that also creates an environment where your stomach or intestine become pervasive, or it blocks the holes between the cells. So no bacteria will go out of the lumen. Basically, they are containing all the bacteria in the lumen itself, so that your rest of the body is sterile, which is very, very important. And when you look at germ-free mice, we have a colony here, like in our animal facility, where these mice doesn't have any bacteria in the body. And then we find that, okay, these mice, they don't have functional immune system. They have problem in digestion. Their body will look different. And the reason is because they have never seen a bacteria in the entire body. So they're not getting a tonic stimuli to create or mature their immune system. So that's why you need a bacteria in your body, but you need the right one and prevent the bad one. So this is very complex figure, right? So what do you focus on this? This is your epithelial cell lining of your intestine, and this area your bacteria resides. With the help of these immune cells that live in the intestine, they communicate with your intestinal bacteria, and they produce a lot of cytokines, a lot of chemicals, and they help a tonic regulation of your health. So when you don't have this immune cells not going to be there, because they are coming there in the gut because of you have bacteria in your gut. And why these are important? If you look at the entire body, almost 70 to 80% immune cells, they live in the gut. Normally we think about, oh, we have blood. If you talk about immune cells, you would think about blood. You would take out the blood and you'll do the immune analysis. The problem with the blood is only a fluid that cells are coming into it. But these immune cells are getting education from somewhere else. And that is the gut is one of the important one. So currently we are working on different kind of products. And one product is a protein came out from this bacteria, which is Lactobacillus acidophilus, which is very, very common. Everybody knows that. Everybody has taken some time of their life because it's one of the bacteria is in yogurt. So it's a gram-positive bacilli, means, and it produces lactic acid. So we call them lactic acid producing bacteria. It's a group of bacteria. And they present in human gut at lower concentration, always, most of times. Most of the healthy gut is available in yogurt. It sustains a lower pH so that when you eat it, it passes your stomach alive and inside your gut, in your lower stomach. And it's considered as probiotic. And a lot of companies sell it, this product. So this is one of the product that you see here. It is from Members Mark, from the Sam's Club. I'm not affiliated with Sam's Club. Actually, I'm going to show you this opposite. It doesn't work. So this bacteria just came out or isolated from human gut in 1975, long back, like 45 years ago. Somebody has isolated from the human feces. And since it came from human, they thought, oh, it must be good because it came from healthy individual. So when we started our work, first thing we did, we induced colitis in the mice. So we took the mice and there's a way to induce colitis in the mice. Details is a little bit long and cumbersome, but we introduce a particular cells from other source of mice that so that these mice get inflammation in the gut specifically. So what we are showing you here is a body weight change and is a weeks of transfer of the cells, inflammatory cells. So when NCK56 is the same bacteria which has been sold, when you gavage them or feed them this bacteria, you don't see any difference. So this probiotic might be helpful to maintain a healthy gut, but when you have a disease, it may not help you. So, so we start thinking about it, why it is happening, and can we improve it? So same time, we and others have figured out that this is the cell wall of the bacteria. So we figured out that this bacteria has multiple things on it, and there are some protein, we call them surface layer protein. As the name suggests, it's on the upper surface. One of these molecules might be helpful, but others may not. So we figured out that there are three proteins on the surface. One is called SLPA, another is B and X. And as I previously, somebody reported that maybe SLPA is a good one, B and X may not do something. So how we can harness this advantage of taking advantage of SLPA? What we did, we start knocking genes out of this particular bacteria. First thing we did was taking out LTAs called lipoteichoic acid, which is a very inflammatory molecule, like a lipopolysaccharide of E. coli. So if you get injected or get exposed to it, you will have fever, like a normal immune response. So first thing we did, we delete that gene out. So now it's gone. Then we deleted SLPB and X. So leaving only SLPA on the surface. So this bacteria, which looks like this, now become naked like this. So what really helps us, now this bacteria is going to show or get exposed, SLPA is exposed. Now immune cell will recognize SLPA much easily. And the response what we'll see is much more profound. And then we will test it whether SLPA is important or not. And this is electron micrograph. So this is what normal bacteria looks like. When you delete all this material, they become a little thinner. Cell wall become thinner. That makes sense because you just taken out the material from the cells. So next thing we did is, again, we went back to mice to test whether it works or not. So, there are few jargons here, so I'm sorry about that, and I will try to explain them. So one molecule is called IL17, interleukin 17, which is a pro-inflammatory cytokine. When you have more, it is more damaging. And other molecule that counteracts this inflammation is called Treg, or we call them FOXP3, is a transcription factor. So bottom line is, when you feed this bacteria, either PBS, the solvent, or the wild type bacteria, or the other bacteria, what you see is not having all those layers on the surface. Now this bacteria is generating beneficial, good cells in your body, which is, we call them Treg, which is very, very important. And right now, like in the US, they have diabetes being treated by this cell itself. So they will take out your body blood, they convert them to this particular cell, and they will put the cell back for treatment of a lot of inflammatory disease. So this is a good cell to have. So now this is a much more interesting figure. So I told you or showed you earlier that this is the weight graph and this is the weeks. So when we transferred or caused disease in these mice and fed these mice either PBS, solvent, wild-type bacteria, or the mutant bacteria, or purified SLPA, we biochemically isolated SLPA and fed the mice, or just transfer Treg, which is a good thing. That is our outcome, right? So in all the cases, what you want to see is, so when you have a disease, you see the purple or pink, dark pink one. So mice start losing weight over time. So they are getting scrawnier and scrawnier. They are not able to utilize the food. They are getting smaller and smaller. And this is the NCK56, which is the wild type, is being sold as a probiotic. Again, as expected, they go down. There are not any difference between the normal, no treatment with the treatment. But when you see this NCK2187, which is green line, you can see that they are maintaining weight. They are maintaining their body weight. They are doing well. Same thing holds true for the simple SLPA, which is the blue line, which is also helping. And also you can see with the Treg, which is going to fight the inflammation anyway. So that's working. So looks like our SLPA protein is able to help or prevent colitis. And this is a colitis score. There's a histological measure. Like after euthanasia of the mice, we'll take out the tissues. Our veterinary pathologists will look into it. And based on that, they will give the colitis scoring. So that's that information. So it looks like the SLPA or the bacteria that has more SLPA on the surface is helpful for colitis. So the problem with this bacteria is, first of all, we did our experiment with the isolated SLPS, isolated surface layer protein. The problem is that protein is, you have to grow a lot of bacteria and get very little. So it's not going to be commercially viable project to do anything with it. So we have to think about some alternative to use them as a drug. Second is, since the parent bacteria, parent Lactococcus lactis, Lactobacillus acidophilus, came from human gut. So there is a chance when we use this new mutant as a drug to a human being, they may again colonize the gut. And we don't want that. We want to use this as a drug, not permanently changing the microbiota of a human. Because we don't know what will happen having the SLPA exposure for the long period of time. It might be cause immunosuppression because we are trying to suppress the immune system by using this molecule. And third is, this is recombinant bacteria. So we have to have an environmental containment system so that when it comes out from a body in feces or some way, it has to destroy itself. It should not just keep living because you don't want to spread this bacteria in the environment. So what we did, the solution what we did is we express this protein, SLPA protein, in a bacteria which is approved by FDA to deliver any biochemicals to the gut. Secondly, we built a environmental containment system so that we can grow the bacteria in vitro in the laboratory, but when we feed the mice or human eventually, then when it comes out in the feces, or if you try to do the dogs, it comes out in the field and it should die of its own instead of being in the environment for a long period of time. So what we did, there is a bacteria, we call them Lactococcus lactis. So this bacteria is approved by FDA. A lot of research has been going on this. And if you go to clinicaltrial.gov and type this bacteria, you will see like at least 10 of different trials must be going all the time using this bacteria. So this is very, very safe. And that is the reason we chose this. And what we did, we expressed this protein. And what we did is we don't want this protein to be tagged to the surface. The reason is, you think about in the gut, you're putting a small amount of bacteria. There are trillions of bacteria already there. So the chances of getting this recombinant bacteria to right cell is very limited, very, very limited. So we try to make this protein more accessible, available to the immune cells. So what we did is, okay, we made this protein to be secreted out in extracellular milieu. So it'll be in the fecal material, it'll just floating around, and your immune cells will pick up. The chances is higher. And when we did that, we got better results. But this is data, is a boring data, just to show you that, yeah, we able to express it in the cytosol and also in media. So protein comes out on the in extracellular milieu. So it will work. That's the idea. Second, this data is we fed the mice and started looking for the presence of bacteria, and we saw that bacteria disappears within 24 hours from the body, whereas the 2187 remains there for at least five days. So it is transient. You feed them, just comes out and gone. And this is again the same numeric, different representation of same thing in using different technique. So it's showing that your protein has been secreted out and it should work. And then the question was environmental containing them to grow in environmental setting. So what we did, we deleted one essential gene that makes thymidine, which is a part of nucleotide that is essential for your DNA synthesis. So if you don't have this, bacteria will not make any further DNA, so they will not replicate. So what we did, so we grew the bacteria in two different conditions. This is media, this is the typical media, and with and without thymidine. Without thymidine, they don't grow at all. So that is in vitro. Then we thought, okay, this is in vitro, let's do it in vivo. Can we see that in the feces? So we took the mice, we fed this bacteria, and after feces came out, we grew them in presence and absence of thymidine. And this is the wild type one, which is the unmutated bacteria. This is the mutated one. And you can see that there's no bacteria is growing, or very little bacteria, if any, is growing. So showing that when we use this bacteria in human setting or in other setting, vivo setting, nothing will come out. Bacteria will not survive in the environment. Just to make sure everything will be safe. And second thing we did is looking for same molecule, Treg, where I was talking about, the anti-inflammatory molecule or cells, whether this particular bacteria, this particular bacteria, which is now we call them R110. R110 is comparable to the old bacteria or not. When we did that, what we did, we orally fed this mice 1 billion bacteria per mouse, 0, 3, 6, 9 days. And after that, 3 days, we just euthanize the mice and look for the particular anti-inflammatory cells. And we see that, okay, now this bacteria is inducing comparable level of this particular protective cell in the gut. So this was very good. So then we did the classical, our colitis experiment. And what we found, this is the same way we cause the colitis. And then we fed this bacteria. So when you have the PBS, like a solvent, you have a lot of mice were losing weight, and eventually a lot of they are so ill that we have to euthanize them. And 2187, it protects the mice, but R110 was even better than the original bacteria. And this is the FOB score, which is a fecal occult blood, because when you have colitis, sometimes you have blood in the feces, and whether that protects or not. So yes, this mice R110, which is our interest now, which really protects colitis from colitis, and also if you look at the blood in the fecal material, that is much less. And these are again jargons, but these are important. So like these are cytokines, which is IL-1 beta, which is very, very common. A lot of people heard of Ankara. There is a injectable drug people use to block this for colitis and a lot of other diseases. So this particular bacteria reduce the IL-1 beta transcription in the colon. And IL17, as I mentioned earlier, which is a bad cytokine, bad immunological mediator, is gone or less. And the good mediators like interleukin 10 has been increased in the gut. So, and again, so what I'm trying to say is now we have less inflammation and more of the protective cytokines and cells are proliferating in the gut. Again, this is the serum cytokine. Now, we saw in the gut, can we detect in the bloodstream? And this is very important because we are trying to take this product to human. So in human, as we know that when you will start this type of work, the only thing will be accessible is the blood. So we need to know biomarkers are available on the blood, so we can say that, oh, this is working. Because like in the mice, we cannot biopsy any individual's tissue, colon tissues. So when we did that, we saw that the similar pattern. We saw this is an inflammatory cytokine. It's called IL-12. And these all are gone down. And the protective cytokine, which is, we call them interleukin-10, which has been elevated with the treatment of R110. We always kept the NCK2027, which is a mutated bacteria of Lactobacillus acidophilus, as a control, so as our benchmark. And then we looked at the tissue, whether there is some, these are called mucin. So you need mucins to be produced from your epithelial cells to keep those gut bacteria contained in the lumen. Because you want food to get digested, bacteria, you need them, but you don't want to too close to their own tissue. So that's why production of mucin is very, very important. So which is elevated by this treatment. And these are tight junction proteins, which is needed. So see, this is, say, these are small boxes, epithelial cells, and anything can pass through between these two cells. To seal this gap between the cells, you need certain proteins. It's called tight junction proteins. So when we treat them with this bacteria, we see the increase in these proteins. So to make sure the junction between these cells are sealed, so nothing is coming out. So it is again to protect our body from fecal bacteria to coming out of the systemic circulation. So that was also working, and this is a typical assay. In human, we also do that. We call them calprotectin test. When you have colitis, this is one of the early event, and we detect in the feces. So basically, we try to keep all the serum albumin in our blood vessels, in our tissues. When your intestine is leaky, when these are broken down, your blood proteins comes out in your fecal material, and that's what we detect. Or calprotectin is the same way. So they are very early events you can detect. When you do that, this is the PBS that has a disease, and you have a very high amount of albumin coming in the fecal material. But with these two bacteria, they are very, very less to know. So suggesting that in all different measures, this bacteria is able to protect from colitis. So now it was probiotic. Now we're trying to use them as a therapeutic. So then we thought, okay, when we have to take this bacteria eventually to human for make it the human drug, and we wanted to know what really this protein does to our immune cells. So one way of looking at it is in vitro, which is much more contained. We can measure the gene transcription or protein expression, all these things very easily, instead of looking into the in organism, because a lot of things are going on at the same time. So we took the blood from the individuals and converted those blood cells into a particular cell type, which is, we call them dendritic cells. These are very, very important immune cells. And when we did that, don't get overwhelmed with all these figures, because what it is if we treat these cells with three things. We left the unstimulated, just left it alone, or we use a wild type lactase. It means this bacteria is not producing SLPA, or the one R110, which one? Lactase, which is expressing SLPA. And then we did this called RNA-seq analysis. So we are looking for all genes, is elevated or down-regulated. what we saw is there's a lot of changes are going on. But if you group them differently, there are cell surface receptor, cytokines and chemokines, which are soluble mediators from one cell to another to communicate between the immune cells. And certain enzymes were elevated, and some interferon response genes were elevated. So baseline is, which were really was striking, is a cytokine which we never thought of, which was IL-27 here. Because others, we knew that these are anti-inflammatory and this will do something, but IL-27 was one of the unique one. The reason behind this, because when we IL-27 is a cytokine which can be anti-inflammatory or pro-inflammatory, both sides it go. But in our model, we wanted to really verify IL-27 is doing something, because as I mentioned earlier, if you treat yourself or a human being with the same SLPA over and over, we wanted to avoid that because we thought it will cause super immunosuppression. But IL-27 is a molecule that will not cause immunosuppression because it also creates a little bit tonic inflammation to your body. So it is beneficial cytokine instead of complete immunosuppression, like I was referring to a cytokine called IL-10. So when I saw that in human, so we wanted to know whether it also applicable to mice, because in mice we can change the mice behavior or change the gene or something, so we can study better in the mouse model. We have limitation with the human tissues. So we redid the experiment of colitis as before, and then we isolated cells, gut cells, and looked at whether this treatment induces IL-27 or not. And yes, it did. So we were on the right path, and human cells and mouse cells both behave same. And this is very, very important because we are trying to model disease in mice of human disease. So they should be behaving same way. Otherwise, we are just studying an artifact in the mouse. So, and then we proved it, whether IL-27 is a key molecule or not. The reason is, we created a mouse, so I'm not going into detail how we created it. Basically, we took this mice, which has IL-27 receptor. So receptor is gone in this mice, and we bred with the rag mice. There are another set of mice to create this mice. So there's a lot of breeding and testing by genotyping to know this mice. And then we provide the naive T cell that causes colitis to cause colitis. So when we did that, we stopped seeing the difference between IL-10 and IL-17. This is the old figure that I showed you. What happens in a normal condition? You see increase in IL-10, decrease in IL-17, and increase in IL-27 in myeloid cells. So, myeloid means cells, macrophage and dendritic cells. We call them together the myeloid cells. So more importantly, you see the increase in IL-10 and decrease in IL-17. Not having IL-27 receptor, it changes the pattern. So what it tells you that IL-27 signaling was important for the phenotype what we saw. So the question is, can we use IL-27 itself instead of doing all this thing for protecting colitis? So there was another group in NCI, National Cancer Institute. They did that. So what they did, they took the Lactococcus lactis, the bacteria we are using, and genetically tried to express interleukin-27 and tested in colitis. It works. And importantly, a lot of people who suffer from ulcerative colitis, they also have a point mutation in this particular gene, interleukin-27 receptor. So it tells me that, okay, that is the important molecule. If we supplement that, it potentially will help. So this is we come up with, like SLPA binds to its receptor that we found earlier. This expressed on dendritic cells. That leads to production of IL-27. That produces IL-10, which inhibits inflammatory cytokines and helps Treg and all this thing. And we also found a metabolite, which is called 3-HHA. We are working on it. We don't have much data on this, but hopefully within a couple of years, we will have data to show that how this particular molecule changes our metabolism to induce this molecule to kill inflammatory cells. But we are just working on it. But what I showed you, I tried to show you a lot of things for the colitis. And it makes sense to start with the colitis because the bacteria we found, it was from gut. And so that it's logical to see whether you have, you find any protection in intestinal inflammation. When we were working on the gut, on the colitis, same time we saw a pandemic, COVID. And then we thought, can we see some protection in the lung? Because we always hear that different kind of variants, maybe vaccine will work or may not work. So we want to know whether it has a systemic effect or not. The rationale of looking at systemic effect, because we saw serum cytokine, the cytokine, the soluble mediator, that is also changed when you're feeding this bacteria orally. So something was happening in the body, something was translating in the entire body. So the question is, can we make our lung better if we use this bacteria or not? So with that idea, we first thing we did, look for the good cell, the Treg in the lung. So we haven't done that before. So we thought, okay, let's try. So same way, same experiment we did. Same pattern, we fed the mice 4 times with the bacteria. And after 3 days, we euthanize the mice, look in the lung whether we have a good cell or not. And we did find good cell in the lung. We found a robust increase in the lung, which was surprising to us at that time. And I thought, okay, let's do something else. So for all inflammation, most of the lung inflammation, we blame a particular cell, we call them neutrophils. So neutrophils are the bad cell. I'm not saying the bad cell. Everything is in context. So neutrophils are the first responders for any infection or any inflammation. They reach to the site of inflammation. But these cells are soldiers. They will find something to kill and then die themselves. So they will ooze out their guts out, right? They will die and like they're kamikaze, suicide bomber, whatever you want to say. That's what they are. So when they do that, they will release a lot of material in the extracellular milieu, which is very, very inflammatory. And if you hear that in COVID, what happened? A lot of people who are suffering from hypercytokinemia, they have a lot of cytokine being released in their body. And one of the treatment like dexamethasone and also anti-cytokine therapy are being used in COVID to prevent that hyperactivation of the immune system. So the first goal was look at whether we can reduce the initial surge of these cells. So same thing we did. Same thing, we fed the mice, and then we put some inflammatory molecule in the lung. Here we is using LPS, lipopolysaccharide, which is a cell wall component of a lot of gram-negative bacteria like Salmonella, E. coli, to mimic that inflammation. So when we did that and we get the cells out and run on the flow cytometer, that's what our bread and butter. So we saw this sharp reduction. You can see the 82.8% of those cells in the lung were neutrophil in this condition. But when the mice were fed before with 110, now you reduce it to 1/4. So that was very striking. So okay, looks like it's going to work in a real condition. So the next thing we did, we thought, let the mice live and see the full course of the inflammation of the lung. So again, we start gavaging, 4th gavage like we did earlier. And then we gave a whopping amount of lipopolysaccharide, very, very high dose. Means 3.75 microgram per gram body weight. It was like, it is, you can see here, means it just within hours, body weight, the next day, body weight of the mice, was 20% less than they started with. So they just reduced, they cannot even breathe. They're very, very in a bad situation. And then we wanted to see what really happens, whether they recover or not. So here is a red line, you see that. When we still the LPS in the lung, they start losing weight and gradually they recover. Normally, like you have inflammation, you clear up the inflammation, you survive. But the one that we received, our bacteria, that lose a little tiny bit, but never lost like this. It was a striking difference. These mice about to die. It looks like they're going to die today or tomorrow. They barely survived, and few died as well. But other group, they survived very well. They had no sign of symptoms of disease at all. So that was very surprising to us. And then we thought, okay, after the completion of experiment, you have to euthanize the animal, look at the lung, what really is happening. Is the good cell is still there? So we saw that good cell that we talked about, Treg, it was still there in the lung. So it looks like having that particular cell type helping the lung to reduce pathology or not cause the disease in these mice. Oops. And we always hear about when you have lung infection, a lot of people have fluid in the lung, and then somebody has to go in the lung and take out the fluid to let your lung heal. And that happens because lung loses the tight junction proteins and causes the gap between the epithelial cells like this. And a lot of blood and blood fluid, not blood itself, fluid from the blood, they will get into the lung to reduce the lung capacity. So we want to see whether it also helping in the regeneration of those cells so that they will pack those the gaps. When we did that, we saw that these cells, which should be damaged, they are replicating more. They're dividing more. When they divide more, what they will do? They'll start packing up to build the barrier so that not more fluid will go inside much more faster. So maybe that is one of the reasons why we don't see that sudden drop in body weight or the symptoms. We are still working on this, but I think this is much more relevant. We got some money for doing lung experiments and finding out the mechanism, and we are going to use this for influenza initially. And we also wrote for the COVID. We got very good response to do the same experiments on COVID in the mouse first. But hopefully within a year we'll get more money. But right now we are doing with the same experiment using influenza. One reason we are doing for influenza as well, because we also saw some interferon response gene. If you remember those crazy graphs, so those genes also inhibit viral infections. So we thought lung infection and viral infection, there's two things very common, is the COVID or influenza. Influenza, we can do it in the very small scale, we can do that. For COVID, we have to go in a different kind of containment facility, and that will be future. And so maybe in a year or two when we'll have more graphs on the subject. So that's all from me. So I told you a few things, which is most important is when we say probiotic, that probiotic might be good. I'm not saying probiotic is bad. Probiotic is good. My mom takes it and she felt the difference. She is diabetic and now she is saying, oh, I'm feeling very good. So probiotic does miracles. But there is a more to harness from the probiotic or from the gut microbiota. And that's my target, is to find out different molecules. We also isolate different kind of bacteria from human units, which really helps. One thing I can just reveal to you, that the bacteria we found that helps in the bone, strengthening of bone. And I was talking to one veterinarian, he said, like, okay, why didn't you give me that to the poultry? Because now with all this technology, chickens have become bigger and bigger, but their bones are not that strong to keep them alive for a long period of time, because their bone breaks. So maybe that will help. This is the small things, but they might be helpful if you want to tweak it a little bit here and there in your gut. We come to know that a cytokine or a biomarker that we are going to use in human for human trials that hopefully will start next year. Again, we know that the effect of SLPA or any probiotic is not limited to your gut. It is systemic effect in your body. It can influence your immunity in the lung. mood, how you're feeling that day, and a lot of different things. Serotonin that we talk about for healthy brain, a lot of bacteria that helps them to induce that kind of serotonin generation. What we know from this SLPA, that it prevents the excessive accumulation of neutrophil in the lung, the bad cells that comes and destroy themselves. That is helpful. And it also increases the proliferation of those cells to create a impersive environment so that fluid will not get in the lung, so that you're going to breathe properly. And this data I haven't shown you. So we are trying to figure out which cell types is helpful. So we found that the good cell that we're talking about, Treg, that was one of the important one to be in the lung to prevent bad cells to coming in. And how it is happening, that's the new question for us. We're trying to figure it out biochemically and by using different kind of cell mice models to figure out what really happens in the lung that prevents these bad cells out, and how we can harness further from that information. Oops. And these are my people that I work with. So it started with this is the actually legend in the Lactobacillus astaphylus biology. Now he is retired. And the second is M. Jade. He's not here with us now. He left UF and now he's in Texas. And Dr. Zimmerman, she is a dean of College of Medicine, and she is a gastroenterologist. So she is the one who helps me for the human trials, getting me human samples and all those sort of things. And these two people, Richard Hill and Justin Schamberg, they are veterinarians. And with the help of them, we are trying to take this bacteria to the dogs, where they also suffer from colitis. And there are other people in the college and around, and these are two people that work for me in our lab. Thank you.
Thank you, Dr. Sahay. Other questions? Yes.
My question is really two parts.
Mm-hmm.
Did this process actually permanently cure the colitis? And number two is, you had said something about not wanting to change the microbiome, and that's why you went through this process.
Mm-hmm.
Why did you not want to change the microbiome if it makes it healthier and the colitis goes away?
Yes, actually, both are very good questions. So, first question that you asked. What was the first question? Sorry, I just. Why do you want to change the gut microbiota? The reason is, in normal situation, when you have colitis, that is good. You change the microbiota and you might be curing the colitis forever, right? The problem is, initially we didn't know that this particular protein is going to induce IL-27, IL-10, or which kind of changes it is going to show us. If you're trying to change the microbiota forever, it's also possible that same Treg, the good cell, is bad during cancer. So we don't want to push the particular good cells that we're thinking of as good, but it goes to that extent that is going to support cancer in your body. So that's why everything has to lie in balance. If you push too far, it might do something different. That's why we wanted a system where you feed the human or feed anyone, and we can reduce the inflammation. And when they are fine, then we can say, okay, not anymore. You're good. You're good on for your own. And what happens, you said gut microbiota, when you feed this bacteria or the protein, your gut microbiota changes. I haven't shown you data because it's going to be very long. We change the microbiota for good. So what happens, immune system has say on the microbiota, and microbiota has say on immune system. So when we change the immune system, naturally good bacteria just start replicating in their gut. And when they come to a extent that it is good and everything is fine, we don't have to feed this new bacteria, and the natural bacteria will take over and keep keep the inflammation in check.
Thank you. Any other questions?
We have this one question in chat, I think, that all of our presenters get asked. It says, do you recommend taking a probiotic? If so, what type?
Okay, so if you're taking a general probiotic, means I don't have a favorite probiotic, but the one I liked, that I told you, like mom, I gave to my mom when I went to India, and she loved it. So I have to buy in bulk. I'm shipping them. So that's why. But that bacteria, that consortium of bacteria, it is not one bacteria. It has lactobacillus, bifidobacterium, and a lot of them. I got it from Sam's Club. It is, they say, I am not supposed to advertise any company or anything, but it is a basic Sam's Club 10 different bacteria consortium, and that worked for her. And I have also seen it for my dog. My dog was has issues with anxiety issues, and I gave him a bacteria called Bifidobacterium longum, which is a very beneficial bacteria, and he is calm, like it's too calm actually. He sleeps a lot now with the bacteria.
So maybe we should just eat yogurt, right?
Yogurt is fine. Yogurt is fine, but see, all yogurt will have different kind of material.
Yeah.
So you can use yogurt is the best, so that it is natural. Just make sure it is a lot of yogurt. They devoid of live bacteria. You want live bacteria. You don't want yogurt and then there's no bacteria there.
Yeah, it has to have the culture. Yes. Yeah. Now let me ask you one thing. This is not my field, so my knowledge is very peripheral. I thought, from my reading, that the bacteria produce certain compounds which are immunologic, and that's where the benefit comes up. But what you showed today was the bacterial wall induces the immune cells to produce the interleukins, which are beneficial. Mm-hmm. So that's new information for me, or at least different thinking. Now, what is the status of this SLPA in bringing it to market now? So
that was my goal. So when we did that, it is a long process. So when we isolated, right now we are filing IND to FDA, and we got some money for a small human trial. So that will start next year, January, February. So if that works out fine, we're hoping that everything will fine. We did all tox studies in animals and everything. Now, if that is fruitful, those patients will be mild, patient from mild ulcerative colitis. And that's why it was needed to know all the biomarker that we can measure on the blood. So when that is done, then we have to think about to engage some more commercial partners. We have one commercial partner, but we have to engage more. So we are in talk with the big pharmacy and pharmaceutical giants. They can fund this because I cannot make this in my room, right? So they have to go for GLP facility. We have a GLP facility in Maryland. There's a company that who is helping us in making this available as a GLP, but they are small. They are doing for a small number of humans. So eventually that has to be sold out of some pharmaceutical company. Okay. Well, that's good for colitis patients. Yeah. So just following up on this. So what you're doing is you have this protein, the surface protein, you're putting it in a bacteria, in another bacteria that will survive passage through the gut and get to the colon. And then the protein is released, and then the protein produces an immune reaction. But the immune reaction would be only if it's a leaky gut, right? So we first used a leaky gut because we wanted to use something on the gut. And now it's working on the lung as well. Yeah. So we will expand our horizon gradually. But when we started, we wanted to see, because we didn't know that whether it will have a systemic effect or not. We just wanted to look at it in the gut because the bacteria came from gut. So we don't want to just far-flung and doing something, it won't work. Okay. But we got lucky that it has a much more impact. Now we are trying to do a lot of different things like biochemically. This bacteria protein is big. Now we're trying to find out what the small area we can use so that for deliver easily, make without a bacteria, we can make it in the biochemically easily, and we can have systemic delivery, by injection or something else. So we are trying to look for all different aspects of it.
Okay, that's, well, it's something to look forward to for patients with colitis.
Yeah, at least for colitis for now. We'll see.
Yeah. You know, the first lecture we had in this series was on Parkinson's and the thing. And what Dr. Gomez Malay said was that it's also the leaky gut.
Yes.
And that produces alpha-synuclein,
yes,
which goes to the brain and develops in Parkinson's.
So, the old saying in India, a couple things like, if your gut is healthy, you're solving a lot of problems for your own health.
Yeah.
And that's what happens when you have such a time bomb in your gut, all the bacteria. If you have leaky gut, bacteria or bacterial material will go into circulation. And when it goes in circulation, you have inflammation. And depending upon which part they lodge in, you'll have disease. Like right now, diabetes, Parkinson's, Alzheimer's, you could say cancer,
colon cancer,
even the other cancers they have seen. And all these things are caused by inflammation. And if you keep, we take a lot of antibiotics over time. And whenever we take the antibiotic, we are killing the good bacteria as well. And so that leaky gut is very, very common in our own body. And we find the leaky gut or a problem in the gut when we have colitis. But having a healthy gut is very, very important. Yeah. So even you feel like, oh, I'm healthy, but maybe there is some leaky gut. When you're like a diabetic patient, they have leaky gut.
Yeah. Other questions? I'm trying to still get it in my head here. Yes.
I also wanted to know, do you recommend drinking kombucha and eating sauerkraut? I mean, are those good probiotics to have?
I don't know about kombucha. Kombucha is also fermented?
Yes.
Yes, anything fermented is good. Even I will give you one example. There was a paper came out long back actually. Because normally when what we are talking about, when you feed bacteria, the assumption is bacteria will produce something and we will react, immune cells will react to it. Those are also very true, and that's what happens. Like butyrate, acetate, all the lactate, they are also produced, and that changes your immune cells. So one thing is, we thought butyrate and all this lactate and acetate was discovered long back when people start doing research in cows. We know that cow is a ruminant and they have ferment everything in the first stomach that we call rumen. And that produces a lot of all this material. So that's why if you want to keep a cow with a minimum expenditure, you can feed them hay and tiny bit of urea. That is sufficient. Because that urea will provide nitrogen, bacteria in the rumen will convert that nitrogen into the protein of their own, and that protein goes in the gut, get digested. So that is the easier way of keeping the ruminant. So that time people have understood that butyrate, acetate, all these things are being produced. And you might have heard that methane generation from the cow. A lot of people talk about methane generation causing more greenhouse effect and all this thing. So all this thing is happening in the gut, and these metabolites are coming out for different reasons. But more importantly, these metabolites also help butyrate and acetate and lactate. They are the first food that epithelial cell eats. If you have butyrate, that is also contributing to your energy requirement of your gut epithelial cells. Though that's why also important. So, so what your question was? Yes, did Grace, I guess. Kombucha. So in the kombucha, so there's a fermented, right? So they are going to have some bacteria in it. They will produce all these things. So the paper I was referring to, one paper that came out for SLE, lupus. And in that one, although they haven't tried in human, in mice, they took the simple water and they changed the pH of the water. The water was a little more acidic. Now these mice, they have a less of the severity of SLE. just by changing the pH. I was confused that time. How is happening? Because if it is a lactate, acetate, I would understand something happened to the gut. But when I looked at paper, they haven't used this organic acid. They use hydrochloric acid, HCl. So maybe there's a role of pH itself that we don't understand. And also, so that acidic food that we think is acidic, like sour food, that might be helpful in general. But if anything is fermented, they're really good.
Well, the trend now is to have alkaline water.
I know.
What does that do anything to the microbiome? I have no idea.
I don't know. I don't know about
because
it.
people are paying money to buy alkaline water.
Sorry. Yeah, there was for recovery. When you do the muscle, like a lot of exercise, for them that is useful, alkaline water. Yeah. Not for general people.
Okay, that makes sense. Yes. Any more questions? Anything on Zoom? No? Julianne? No? Okay, thank you all very much. Next week, our speaker is Dr. Christian Jobin. He will talk on intestinal microbiota, the barometer of health and diseases. So please come. I'm told he's an excellent speaker. So international.